So let me introduce Hassan. Hassan, I'm going to make first, you understand Portuguese. I'm going to make a short presentation in Portuguese, and you can understand, and then I do the same in English, so that we can start. Bom, pessoal, bom dia. É um prazer muito grande estar aqui com o Syed Shah Hassan. Para quem não sabe a história, o Hassan foi um dos bolsistas CAP Fiocruz CDTS, assim como o Paulo está sendo, como o Carlyle e outros. E ele entrou na última, atendendo o último edital de avaliação. Ele atendeu o perfil de computação, enfim, biotecnologia e computação. E para mim foi uma surpresa grande ter ele como candidato, eu não conhecia. E ver um paquistanês que tinha tido o PhD na Universidade Federal de Minas Gerais e, portanto, falava bem português, para mim foi uma surpresa grande. Eu disse, bom, vamos segurar esse candidato, né? Fiquei muito contente com a aprovação dele no edital. Só que, assim que esse edital fechou e ele foi aprovado, ele teve que voltar. Ele estava em Minas Gerais, mas tinha que ir para o Paquistão para pedir um visto de entrada no Brasil novamente e ficar aqui conosco. Só que aí veio a pandemia. E aí não tinha viagem internacional, não tinha nada. Então, tudo bloqueado. Por outro lado, o Brasil também foi um dos países mais afetados, né? Eu fiquei 3 semanas numa UTI aqui no Rio. Tive perto de ser entubado. Meu médico não sabia dizer se eu ia escapar ou não. A volta foi complicada. Todo mundo foi afetado. A Fiocruz passou a fazer só trabalho presencial. E o Hassan foi ficando no Paquistão. E a gente, enfim, tentando trabalhar com ele desse lado. Como depois ficou claro que uma volta dele ia ser complicada, a gente concordamos que ele ficasse no Paquistão até o fim da bolsa. E esse seminário é uma primeira oportunidade que a gente tem de ouvi-lo ao vivo e a cores pelo Zoom, saber um pouco do que ele tem feito nesse período, os planos dele para o futuro, e depois, quando a gente chegar na question and answer, discutir um pouco que possibilidade a gente teria de continuar a colaborar com ele. Ou ele passando um tempo aqui, ou alguém nosso passando um tempo lá, ou fazer uma parceria Fiocruz, Universidade do Paquistão, para a gente ter algumas metas em comuns. Ele também tem interesse em colaboração com a China, a gente vai ver isso na discussão. Okay, let me now change a bit for our colleagues in Pakistan who do not understand Portuguese. For me, today it's a big pleasure to have Professor Hassan with us, make this seminar. And let me tell you a bit of our history. Professor Hassan, he was a candidate to become a fellow, to get a fellowship from CAPES, which is a Brazilian agency for teaching and university training. We had an agreement between CAPES and our institution, and we are attracting students at the postdoc level. One of our call for applications was in the area of biotechnology and computer analysis, and Hassan was the first name in this selection. So we are very happy and surprised. We are not expecting a Pakistani candidate that had done his PhD in Brazil at the University Federal of Minas Gerais. So it was something of a very unexpected candidate. And from my time in Geneva, I saw how interesting it is to have a team, a multinationality team. When I was in Geneva, I had people from British, Denmark, South Africa, Burkina Faso, Japan, Thailand, Malaysia, several countries from Africa, and it was a very rich environment. I enjoyed very much the 5 years I stayed in Geneva, having people from all over the world. So when I saw someone from Pakistan, I said, well, that's very interesting. Let us get someone from Pakistan. It's not so common. And so we made all our efforts together with the management of the program to keep Hassan there. Even with difficulties of the pandemics, he could not travel, he could not get his visa. But so today it's a nice opportunity to hear in this Zoom conference what has been doing, and later on the question and answers, we can discuss his and our future plans. So Hassan, thank you very much for agreeing to make this seminar, and
Thank you, Professor.
the floor is yours.
Thank you. Thank you very much for such a nice presentation. My voice is clear? It's all right?
Ayesha? Who is?
Okay. Everybody hear me?
Ximena, a gente só vai aceitar a discussão depois. Pode baixar a mão. Tá? Não vai ter discussão
agora. Não baixei
agora.
a mão. Eu sei, dei um ok que o áudio está bom.
Tá.
Okay, thank you very much, Professor Carlos, for a nice presentation, and a very good morning to all of you who are in Brazil, in that part of the country. A very good afternoon to people who are here, because it's almost 6 p.m. In the evening. So good afternoon, good morning, bon dia, bonjour for everyone. So it was nice to have a non-Professor Carlos Morel and working remotely on some topics, and for giving me the opportunity to speak here today. So the topic I'm going to talk on, it's related more to the area that was like part of the work that we have done so far, and some of them it's completed. Some of them it's like I can say 75% of the work it's done, and a little bit it's we have submitted some papers, and it's they are accepted, and we are going to have some good news soon. So with that, I'm going to start. The topic for today, it's a drug discovery against emerging antimicrobial resistant pathogens using genomic data as a tool. So this presentation I prepared for the Centro de Desenvolvimento Tecnológico em Saúde, or Center for Technological Development in Health, Fundação Oswaldo Fiocruz, Rio de Janeiro, in Brazil. My name is Dr. Hassan, and I'm an assistant professor at the Genome Research Center of ICCBS. I'll come back and will explain a little bit, highlight our center as well. So before going to start the scientific topic, I'm going to just give a small highlight of where we are living, where the country is, and how it looks like. So it's if you look at the global, you can see it's a tiny country right there at the southeast part of the world, South Asia, and having an area of almost 800 square kilometers, with the capital Islamabad and a population of around 250 million as per the consensus of 2023. The capital city Islamabad and all the 5 capital cities from Peshawar to Lahore to Quetta to Gilgit-Baltistan and Karachi. Karachi is the place where I'm working and where I am at the moment. So this country, if you look at these pictures, it's I can say it's a gift from the nature, because it has some of the famous mountains, the Karakoram, the Himalaya, which has the K2, the Gorbin Austin, and which has some very barren, without any vegetation mountains. There are deserts, but there are forests as well. There are plains, there are rivers. So it's a variety. It's a natural diversity. You can find 4 seasons here in this country. Right now it's summer, and summer it's like in full bloom. So the temperature is like it can be 40, 35, 45. That's the range. So depending upon the city, the location, it varies. But still, there are some parts even now they have that like the people need blanket for sleeping. So apart from this, the country, there are so many things that I just put a few things from sports and food, because these are the two areas that I like too much when it comes in sports. It's always the cricket between Pakistan and the other countries, especially the India. So it's like Brazil versus Argentina in soccer. The two countries that are crazy for this game. The national game here, it's the hockey at the second, and we have some very good players in the snooker as well. And among the food, as we can see, the very famous biryani and pulao, and the barbecue, mutton and fish and beef. So these are just the few glimpses, not all. Of course, there are so many other things as well. So coming back to the city of Karachi, it's a city located at the Arabian Sea. It's a coastal city, a metropolitan city, one of the largest in the world. It's the biggest city in the country, having a population of almost 20 million people. And it's the 12th largest in the world from population. Of course, this is like Sao Paulo, because it provides the financial assistance, the economy. It's totally dependent on the economy of this city. And it's a very good place for tourism. It can be accessed through air, through land, and through water. So there are beautiful historical places in Karachi, near the Karachi, and around, and numerous islands that are located here. In Karachi, where I'm working, that's called the ICCBS, International Center for Chemical and Biological Sciences. And it started, the foundation of this was laid down in 1966. So it's almost we can say 60, 70 years almost. And here is the person standing right behind. That is the one of the founding father of this institute, the Professor Dr. Rahman, which is famous internationally in organic chemistry, in synthetic chemistry, and natural product chemistry, and who is now the patron-in-chief of this institution. The sitting one here with the glasses, this is Professor Dr. Iqbal Chaudhry, the director of the institute, is a wonderful scientist, wonderful man who is leading the institute towards further progress and development. Now, the Institute of Chemical and Biological Sciences, it has a number of buildings. So it's because it started with the organic chemistry. So the foundation was from the HEJ Research Institute, Hussein Ibrahim Jamal. It was a donation from those people in 1996, and the university provided some land and they constructed the buildings, and some fundings were provided by some other sources. And that journey started at 1966. And at the moment, we have state-of-the-art facilities like the Dr. Panjwani Center for Molecular Medicine and Drug Research, Professor Dr. Rahman Laboratories, Industrial Analytical Center. We have our animal house for the experimentation. We have the National Science Information Center, the Biotechnology Wing, the Jamil Rahman Center for Genome Research, where I'm here working, the bioequivalence studies, the Nanotechnology Center, which is working very efficiently in a very good way, National Institute of Virology, and the ICCBS Technology Park. So it's during the span of these 30, 40, 50 years, the Institute has progressed, and it has the recognition, international recognition. It's part of the TWAS program as well, and it's part of the COMSTEC program as well, representing the country at different international forums. The Jamil Rahman Center here, it has state-of-the-art laboratories for microbial genome sequencing and human genome sequencing and plant genome sequencing. We have molecular biology setup, which is required for the sequencing projects and the genome sequencing, some of the latest, which we are trying to acquire more. Of the available, we have the Oxford Nanopore technology, the MiSeq, HiSeq, the NextSeq, And I have just word from our senior professor that we have a project approved for the NovaSeq 6000, which is a highly efficient sequencer, and will join the team very soon. So in our group, we normally work on human genetics, plant genetics, and microbial genetics. So it's like 3 groups we do have in human genetics, cancer, leukemia population, and plants for the development of genetic modified agriculture-based product to meet the requirement of the growing population of the country. And in microbial genetics, we are working on probiotics and both the pathogens that are causing serious infection. These include virus genome sequencing, bacterial genome sequencing, parasite genome sequencing, and now because I won a project on the fungus, so I can say fungus is also included in this arena. Now we know that currently the established technologies that are in the market. The story started in 2007 by Illumina Solexa, and even now, more than 90% of the world genome sequencing, the projects are performed by the Illumina sequencer. They have the ISEQ technology, they have the NovaSeq technology, they have the NovaSeq X, and their prices, of course, vary because some of them are the compact benchtop for small and rapid analysis, and some are for larger, like human genome projects, and which are expensive, but the efficiency, the output is very, very high. Now, the latest one, the NovaSeq, which has a redesigned flow cell and improved chemistry and optics, it produces 3 times data more than the NovaSeq, the last one, which was here. And they have claimed that they can sequence the human genome for up to $200. Now, there are other alternatives in the market. One of them is the Chinese genomic titan from the BGI. That is the TNP-seq platform, the MGI. It's although it's time-consuming and labor-intensive, but the data obtained, it's really, really good, as described by the authors in one of the work. There is another one that is the G4 benchtop system for smaller and more flexible projects. The list goes on and on. We have in the market Ochima Genomics, and they have claimed that they can sequence the human genome for up to $100. On the other hand, there are Element Bioscience and PEC Bio, which has a quality accuracy of Q40, which means 1 nucleotide error in 10,000 bases. So the accuracy is, of course, amazing, but it's really, really expensive. Now they have brought another instrument, that is review instrument, which cost almost $800,000, and they have claimed that very good data of the human genome can be done for only $1,000. So normally these sequencer, we know that there are 2 main ways where they can be used for production scale instrument, like large scale human genome projects or a single-seq RNA-seq studies, or they can be used for smaller bench type instrument, like we are normally using for the bacterial and the parasite and the fungal infections. And it depends. The cost, it's totally depends upon its use and the quantity of the data that is produced. Oxford Nanopore also have a sequencer that is MinOin, which the whole setup, it's a small one. It's like a lab sequencer. It costs only $1,000 for short reads and for long reads as well. But the other one, that is PromethOin, which is high for performance and produces almost 14 terabyte of data in 3 days. They have launched another one, the P2 Solo system, which costs almost $10,000. So if you look at the price, the Oxford Nanopore is quite ahead in the market, but definitely the amount of data it produces, it's limited. So Then there are some information about the chemistry of the PacBio and the ONT. I'll not go into detail of that. We all know how it works. So all these sequencer, there may be some others, but the one that I just mentioned from PacBio, from Element Bio, from the MGI, BGI, Chinese, and the Illumina, they are the ones which are now dominating the market. And the amount of data, where it is from prokaryote or from eukaryotes or from archaea, there are 3 gene bank databases, DDBJ from Japan, from NCBI, and the European Bioinformatics Information Center. So all of this data is stored in these 3 databases. So if we look at the right side of the slide, we can see that because once you have the nucleotide data, you can have the RNA data and you can have the protein data. You simply need some program, some software to transcribe and translate the data. So a detail of all the databases, it's given here. So it's a huge amount of data that is produced as a result of these sequencing technologies. So if we look at the, for example, GOLD database, which is the Genome Online database for nucleotide, it imports most of the data from the NCBI, and we can see that the number of strains at the NCBI from prokaryote, it's almost 520,000 bacterial genome have been sequenced, prokaryotic genome have been sequenced so far. There are other information. It can be accessed easily from the GOLD database, which is working in collaboration with the Department of Energy, United States government. So this shows a few years back, this data was like in 200, but now we can see it's like in the 500. So this shows the rapid increase of the data. Now, all these data that we see, we study them at 3 levels. One is the structural genomics, the other is the functional genomics, and the next is the comparative genomics. So from the structural genomics, we perform the sequencing, which is the part first that I have just mentioned, and the sequence annotation and the mapping of genes with the related genomes. Then in the functional genomics, we study the transcriptome and the proteome and the metabolome and the other omics that comes under the umbrella of functional genomics, and the comparative genomics study. So these are the 3 levels of studies that we use for all the data that is obtained from the sequencing technologies. Now, one of the part of the functional genomics, that is subtractive genomic approach that we use normally for the identification of, for example, a prokaryotic, a bacterial pathogen, which is causing infection. So how we can find some targets of our interest, whether it's a drug target, whether it's a vaccine target, whether it's a adjuvant target, whether it can be a diagnostic biomarker. And because almost most of the drugs, it's not working against the antimicrobial the pathogens. The resistance is increasing day by day. So it's making a very big problem. So we use the subtractive genomic approach, where we can subtract the pathogen genome to see which genes are essential and which are not homologous to the humans. And in this way, we can obtain a minimal set of genes which are essential and which are non-homologous to the human. So if I just summarize, first of all, the core genome or the proteome is obtained. Then it is compared with the database of essential genes, which is a database that has the genome set, the essential genes from almost 20 bacteria, if it's available and can be accessed online. And then we have to compare the data, that core data, with the host homology to avoid any cross reaction in the humans. So that can be accessed from the NCBI as well. You have to go to the BLAST-P software and then compare your data with the human genome, so that to remove all the sequences that are homologous. And now through that, we can get a set of genes. For example, if the number of genes in a bacterium is 2,000, then after comparing with the essential genome database and with the human, it reduces drastically. So we obtain a set for these targets. Now, the next question comes whether if we are going to use this data set for the drug purposes, do we have the 3D or the three-dimensional model available or not? And what to do next? So the 3D model or the protein three-dimensional structure, it's very, very important when we are talking about the function of a protein. So if you have some novel strain or a strain that have mutated genes, and if you have the gene sequence, then you need to have the protein. So there are information available on the protein data bank. And if we look at the top right, top left of the PDB, we can see that almost 200,000 protein structures that are experimentally determined. They are available on the PDB database. So in comparison to the number of nucleotide data, the protein data is not that much because of the lengthy and the process that the people use to determine the structure, whether it is the X-ray crystallography, whether it is liquid NMR, or whether it is cryo-electron microscopy or other technique. It takes a lot of time, but of course they get a very good structure. So comparing the two, the nucleotide data, that is the huge man, and the protein data, that's the small one. So all the time we need to have some alternative for finding the three-dimensional structure of the target of interest. So this slide shows the experimental structure as well as the other methods by which we can determine the three-dimensional structure. So we have the comparative homology modeling, which is a type of the computational identification of protein three-dimensional structure modeling, and the sequence identity for building such a model, that is as minimum as 30%. So the model obtained has an accuracy from 1.5 to 3.5 angstrom, and we obtain a very good model in some cases, depending upon the sequence identity and other parameters that we use for identifying the structure. We have some other method, which is a threading technique in the case of iTaser, and we have Genovo or ob initio prediction, where you don't have any template structure, so you have to draw your structure from the scratch. Now, for the comparative homology modeling, because it gives a very good structure, the steps, they are very simple. If you have a single a gene or a protein sequence and you want to predict its structure, you start by taking that sequence and identify the related templates. Based on the identity scores and other parameters, you select the template, align the sequences, build the model, and evaluate the model. For evaluation, you have the ProCheck, the SAVE, the ERAT, and other tools that you can use to check whether the constructed model, the quality is okay or not. If it's okay, then go ahead. But if it's not, then come back and select another template and go on until you have a very good structure. Now, the question comes, if this is the process for identifying a single protein. Now, what we can do if we have to identify multiple structures for multiple proteins? So can we do that simultaneously at the same time in a very short time and very good models? Do we have some program? So there is even when I was in my postdoc, there was a software that was developed by the LNCC, that is the Modeline, and it's a very good structure. It uses the inbuilt modeler software for identifying the 3D structure. And if we look, it can identify almost 2,000 protein. It can construct a number of structure in less than 12 hours for a genome having 2,000 protein. So it's highly efficient. This is the main page of the Moline server from the LNCC. I have worked with these people. It's in Rio de Janeiro. And once you have the three-dimensional structure of your protein, then comes the step of the of finding a drug molecule for that protein. So because nowadays there are millions and billions of compounds that are available, and they are ready for purchase, and they are ready for testing in the laboratory. So how we can handle these things? We perform the virtual screening analysis, a large-scale virtual screening for the ligands. Then when we obtain some interesting hits or the ligands, we perform the docking analysis, which is the orientation of these ligands in the cavity to see whether it fits properly or not. And then it is followed by the molecular dynamic simulation studies to check the physicochemical and thermodynamic properties of the complex that is formed. So it's just a general representation that you have a protein of interest, you have a ligand library, then perform the virtual screening for the ligands. It performs different kind of orientation. Even in the screening process, it has a protocol for optimizing the protein structure and the ligand library and correcting the number of hydrogens, addition and removal of bonds and these things. You perform this, and the structure which fits the best, they are the ligands which fits the best in the structure, they are proceeded for the in vitro lab. So here at the moment, because if we have many students here as well, I would suggest that there is a course, 6th Advanced in Drug Designing Workshop 2023. The link is there. It's a very good course for the drug discovery studies. Now, once you have the target identified, then there are few steps that are we can see here. Computer-aided drug design. Of course, you need to have a protein structure. Then you have performed the ligand-based drug design. After that, we perform the molecular dynamic simulation. The structure-based drug design is also, or a lead-based, where we can use an old ligand to identify novel ligands from a library of thousands or millions of compounds. The next step is the ADME reduction, which is the absorption, distribution, metabolism, and excretion of the selected ligand. And finally, we check the QM, quantum mechanics, and molecular mechanics studies. So in this slide here, it's a protocol that I have used, and many people have used with, and I have adopted in few studies with little bit modification. It shows that how one can obtain the genomic data after the sequencing of a bacterial pathogen, and then following different steps to obtain the final targets. In this case, we have 4 targets selected from a few strains of Salmonella typhi. For example, I will show in the next slide. And for those 4 targets, we performed the virtual screening, the molecular docking, and the MD simulation studies. The complexes were then forwarded for pharmacokinetics studies as well. So if we look at the number of the pathogens, this protocol serves almost for all these pathogens that are known as the escape pathogen, which includes the Enterococcus, the Staphylococcus, the Klebsiella pneumonia, and the other Pseudomonas aeruginosa, Acinetobacter baumannii, etc., which are, some of them are gram-positive, some of them are gram-negative. It's that protocol, the steps that can be used the same way or with little bit modification to apply on these pathogens. I'm not saying that that's a perfect protocol. Of course, there are other different protocols. People are using a little bit different steps as well to see how they can determine the structure. So one of the work that I have performed recently, among other works, it was to find the some novel targets, some novel drug inhibitors for the targets which are already reported or which we have reported for the first time in the Salmonella typhi. Because Salmonella typhi, we know that it's a waterborne disease, and even now I have some information that the typhi, it's emerging again, and we have some reports where it's resistant to all kind of drugs, the cholesterol and the polymyxin B, which are the last resort for the gram-negative bacteria, which targets the surface molecules of the Salmonella. Even it's not working. So it's a very hard, and again, it's emerging and emerging. But I hope to have some more sequences in the near future. But last time we have a sequence that's given here that has been sequenced in Pakistan here, and we compared this sequence with the other strains in a test case like 7 more strains. Of course, we can compare it with dozens of other genomes, but here we wanted to see what is the difference between the Salmonella typhi strain, which was so resistant and which has caused so many problems in the area that has poor hygienic or poor other health conditions. So out of the 8 genomes, the 4 targets that were identified, I'm showing here only the result of these of this study, where we can see that the 4 targets after the gene essentiality analysis and the core genome analysis and the homology analysis and the pathway analysis and the protein-protein interaction and other pathways, we come up with these results, which were which were tested for the pharmacokinetics, that the drugs we identified using a library of 12,000 compounds. We had 3 libraries. In this case, we used a zinc library. This library had drug-like properties and it's ready for purchasing. So all of these compounds which were selected, the inhibitors which were selected against the 4 proteins of Salmonella, it followed all the pharmacokinetic parameters. For example, the Lipinski rule, it didn't violate any rule here. And the consensus log P O by W, which is the N-octanol by water comparison. So because the Lipinski rule of 5, it's one of the basic requirement for any inhibitor selection, which explain that if you select a compound, it shouldn't have a molecular weight of 500 gram per mole. It should have a surface area of less than 140. The rotatable bond should be less than 10, and the hydrogen bond acceptor and donor, and in the same way, the hydrophobicity and the lipophilicity. So if a compound is fulfilling this criteria, then we can say theoretically that yes, it can be a very good inhibitor. So these compounds were then, after the pharmacokinetics, their molecular dynamic simulation studies were performed, because this is one of the requirements if you are performing the in silico or the computer-aided drug design. And these MD studies were based on a free binding energy calculation. So we can see that the free binding energy calculation here in the table, they are all in negative, which means that the ligand protein complex that is formed, they are stable. The other parameter for the MD simulation that we did was interaction calculation. So we can see here that the orange color, it represents the hydrophobic interaction, the blue color, it represents the hydrogen bonding. So we can see that the A, C, and D complexes with their inhibitor, it has these complexes are stable because of the maximum hydrogen bonding and hydrophobic interaction. In the case of B, we can see that there are some other forces like salt bridge, pi stacking, and pi cation, which stabilizes the ligand protein complex. After that, the other 3 parameters that were performed for the MD simulation analysis, that was RMSD, which is the root mean square deviation. It shows the deviation of the original protein structure and the structure which is modeled, and then the ligand protein complex together and deviation from its original structure. So we can see the RMSD values that are that shows a little bit variation in the beginning from 0 to almost 75 or 100 nanosecond. But after that, for the rest of the 200 nanosecond, it attains the stability. The same was the case for the RMSF, which is the residue fluctuation, and the radius of gyration. So in maximum cases, there are some residue which are showing fluctuation, which are interacting more, but overall, the result showed that the complexes are stable. And because we have, if we see here just quickly, for each protein target, we have selected top 2 hits. So in the case one compound is showing variation, we have the other ligand for testing as well. So that shouldn't be a problem because we always go for that. Now, I think this slide is a little bit early. It needs to be at the last one slide, but the virtual ligand screening or the VLS that we call, it's doing a great job, and because of the availability of the number of compounds that are available, we can see here, for example, the red line here, as the number of compounds are increasing from 1 billion to 100 billion to 10 billion, and if we look at the left side of the graph, we see that it gives the best scores. So now, in our case, we use small libraries, but in the case, if you have a very strong computing system and the setup is there available, you can perform the large-scale virtual screening as well, and this will give the chances to obtain some ligands with very best scores. Now, the blue curves on the other side, we can see that it shows the percentage of the predicted hits validated. For example, how many hits that have been identified through virtual screening, they have been already validated experimentally. So we can see that as the number of screening increases, the number of this percentage also increases. And it has been done for 100 nanomolar, 1 micromolar, and 10 micromolar. So they have been checked with the literature as well. This table here, it's a bit small one, but I didn't have the enough zoom. So this shows some of the some of the softwares, the program tools, and the algorithms that are used for the large scale virtual screening. Now, this slide is very interesting because it shows a symptom-based hierarchical screening. And for example, in this case, they have selected a sulfonamide drug, which has two, for example, alkyl groups R1 and R2. So what they did, they actually kept or covered one functional group on the one side, and the other one was enumerated or selected for the screening purpose. Now, if you look at the bottom, for example, here, it's a protein binding cavity, and the sulfonamide drug, it's there, where we can see that one of the functional group R1, it's kept, while the R2 is shown. Now, the R2 represents 3 different structures. If we look at the left side of the figure, R2 has a triangle, a rectangular, and a pentagonal structure, which means it can be a different structure. It can be a benzene, a phenol, or any other structure, and changing one by one this R group, and then performing the virtual screening. So if we come up with the 4th step, where they have tried the different structure in the R2 group and then replaced also the structure in the R1 functional group, we can see that out of millions of compounds, they have selected 100 to 500 candidates for experimental testing. So this is how the virtual screening works on the larger scale. This figure, I think it's the second last figure, which we can see that if we have, for example, the availability the computational resources and the availability of the compounds are already available there. So it shows a comparison of the computationally driven ligand selection and the experimental driven ligand selection. In the upper part, we can see that how long it takes. The timeline is from 2 to 12 months if you have millions and billions of compounds towards the drug candidate. While in the case of classical high throughput screening performed in the in vitro studies, it takes almost 4 to 6 years for the drugs to select a drug candidate, whether it's a direct one or it's a lead-based candidate. So this of course shows the high throughputness of the type of studies that are performed using the computational screening of different ligands. And as a conclusion, just the order was because these things have become possible because of the 4 main reasons that are given here. Because we just saw from the PDB database that with the passage of time, especially with the increasing number of 3-dimensional structure determination techniques, the number of protein targets, they are becoming available more and more. So there are abundance of 3D templates present. On the other side, the factor that is the growth of virtual chemical space or the number of compounds present in the public databases for large-scale screening, and there are studies reported that they have obtained very good results with this computational-based screening, where they didn't need to synthesize some new compound, but they just performed the screening and they identified very novel and very potent inhibitors. The other factor that is making it very successful is advanced computational methods. The computational methods, every day we see that there are advanced and robust computational methods are coming in the softwares are developing in the market. And the fourth is the accessible computing power. For example, 10 years ago or 15 years ago, there was a problem, but now you have the clouding system and you have the accessibility towards the high throughput and the servers and fast servers, where you can sit here and perform your analysis on the other part of the server. So all these are the main reason which has made possible the high throughput, large scale screening of these compounds, and which is very good because we can see that the percentage or the ratio of the resistance of microbes or the pathogen that is increasing. Of course, this computational power, with the increase of this computational power, it's coping with the problem that we are facing. Now, I would like to acknowledge some of the people here. Of course, that's the Higher Education Commission of the Pakistan, our patron-in-chief, Professor Dr. Atar Rahman, our director, ICCBS, Professor Dr. Pal Choudhury, for giving us the opportunity to work here, senior advisor, Professor Dr. Shahid Mansoor, and I forgot the name here, Professor Dr. Carlos, but I'll mention together with the picture as well here. And this is the team from left to right, Dr. Irfan, who is a very good bioinformatician, and all the problems that we are facing in the bioinformatics. Of course, we sit together. Dr. Asad Karim, a wonderful bioinformatician as well, working on the microbial pathogens. Dr. Irfan is working on human, and sometimes I drag him to the microbial part as well. The third is Dr. Shakil, which is working on cancer, genomics, leukemia, and these other. That in the center, that's me. Professor Dr. Shahid Mansoor, who is a senior professor with a lot of publication and citation. He has recently joined our group as a advisor, and everything that we are performing here, for example, the projects and the planning, that's only and only because of his guidelines and advice. And the final, it's Professor Dr. Ishtiaq, who is in charge of the Genome Center. Down here, there are some girls, some beautiful girls sitting there, our students who are working all the time and who are always available for any kind of research experiments that we needed. In the work that I have done so far, we have published from that program 3 publications so far. The 2 of them they are accepted, and we are waiting for the final hearing from that side, which certainly I guess will take a month or 2. And here are some areas that together with the doctor or my team members that we have planned, and I was thinking to discuss with the Professor Carlos Morel and the other members here as well. So thank you very much for the time and for the attention. If there are any questions or if there is something for discussion, I give the floor to the Professor Carlos.
Thank you very much, Hassan. Nós vamos abrir para perguntas. Quem estiver na sala Zoom, é só apertar o ícone da mão ou escrever no chat. Quem estiver no YouTube, é só escrever no chat. Any question by the people in Zoom room, please press the hand picture or write in the chat. Or any question by the people in YouTube, only write in chat.
Okay, Hassan, please stop the sharing of screen.
Okay.
So that we can go back to the good.
Okay.
Eu vou só fazer um comentário, Carlyle, antes de você falar, tá? I think it was excellent seminar. Congratulations, Hassan, because in addition to the science, you gave us a flavor of what is the country, Pakistan, with all its beautiful cities, food, and the buildings, and the settings and scenery. I don't think that, I think no one from our center has gone to Pakistan, so it was very nice to see this background. Second, I think you gave us a very nice picture of not only of your work, but the work, the atmosphere, the buildings, and the kind of international relationship that you have. I think that was very interesting. I was also amazed with the comparison of genomic sequencing machines and hardware. It's quite an exercise these days, because almost every day you have either a new instrument or a new fight, a new patent disputing, and it's not easy to navigate in these waters. So thanks again, and I think it will take us a lot of time to swallow all the data that you showed us. Okay, let us move to questions, and my colleagues Claudio and Marcos Vinicius will see who is next on the line.
Okay.
Carlyle, vai lá.
Hassan, congratulations for your presentation. So I have some questions about your job, specifically on the free energy. You prefer to make the questions in Portuguese or in English?
I guess we have some students from here and there, so if it's English, it will be better. Otherwise, I have no problem with Portuguese.
Okay, so my question is, when you make the free energy, do you compare the structures, because the method of AMR-PBSA is not very good results when the ligands is so different. My question is, do you compare the ligands before you make the AMR-PBSA? Or do you usually this ligands suggest to be your visual screen?
It's always good to compare the structure individually before performing this screening, but because we have used this and we have this already the team members who are working on, they have a hand-on experience on the CHARM platform, the NMD. So in this case, I guess they didn't compare the ligands before, so they have performed for the together for the ligand and in the complex structures.
Yes, because in your table the surface of the ligands is so different. Have ligands of the 26 of surface, others is suggesting the large difference between these ligands.
Yeah,
so
that's very true.
I'm suggesting you make it very careful before you make the free energy.
Well, in fact, we have. I'm sorry to cut you in the middle. We have, in fact, very almost the same kind of comments from the journal from one of the reviewers asking these things, and even he asked for if we have performed these free energy analyses and the RMSD and the RMSF and the RG and all these things, if we have performed for the apo structures, which we didn't, of course, but I must say it's a very valuable and very good suggestion, and definitely I'm going to talk to the team members to be careful about it in the future.
This my second question is the windows of the make the free energy. You just use 215 of the windows for the free energy calculation. So you not consider to improve this to windows because the free energy methods is so sensible. So if you do improve the windows, it's possible you to make more accurate results. If and after you could compare to experimental analysis. So the Windows is so smart. So you make it careful to improve the Windows in next work, or do you continue to use the same protocol?
Well, we do have a specific protocol, and if we just come back to the slide, I think I have stopped sharing. We have these two guys from here, and we have one partnership from Brazil who is performing this the MDA simulation part for us. So I think I have to chat with him again about this question that you have raised to perform, because the next stage definitely that will be the testing of the ligands. So it all depends upon the data that we have obtained here, like which one ligand to select for the test and which one not to select for the trials. So you're right. We'll check this.
Okay, thanks for the conversation, Hassan. Congratulations for the presentation.
Thank you very much.
Thiago.
Thank you, Marcos. Thank you, Hassan. Congratulations on your presentation. I was particularly interested in one of your final slides when you compared your computational approach to the classical approach, and the time frame you have, the shorter time frame you have to get the results. And that's very important for the discovery and innovation process. With Nicola and Carlyle here in Rio, we are trying to do a similar approach to guide some compound synthesis and make some modification, well, to guide the medicinal chemistry. But one of the things I was wondering when you presented was how accurate, how could I get a sense of the accuracy on the data you generate? So some of these hits you identified, did you test it in the specific enzyme assay or against the salmonella or the microorganisms? You identify the hits so you can have some kind of orthogonal results to reconfirm that the hits you identified by the computational biology approach, indeed they work in the microorganisms or on their enzymes. So this is one question. The other question is, if you take a very known and studied enzyme, let's say, for instance, the SARS-CoV-2 major protease, which is a very important enzyme, and new drugs have emerged, many different works on drug repurposing, but we do have a good compound, which is nirmatrelvir, that makes Paxlovid, the compound that's used. So if you target this protein under your computational conditions, and among the drugs you are testing, you include the nirmatrelvir, will it be the most significant hit or not? I think that's another way to try to get a sense on the validation on the data you generate. So this is the two things that came to my mind when I watched your presentation. One, to reconfirm by enzymatic or infectious microorganisms assay, and the other as a non-control on enzyme and target on your computational pipeline. But thank you again for the presentation.
I got your both question, very interesting question, and that's of course the type of questions that really we need to address even in the paper. And of course the scientific community wants, if I selected and I propose some drug inhibitors, some drug molecules as inhibitors, whether it will be working or not. So I will answer in the way that in this work we have tested like 12,000 compounds, and we purchased these compounds, and I think it's just arrived. And not only these compounds, because I have another library of compounds which is from here, because in our institute we do have some very good scientists. So what I'm doing, I will do, because I was waiting for the salmonella, the one that we have sequenced the other year. I don't know if it was right there or not, but now I got the typhi strains, I got the acidobacter strains, and I have like 15 or 20 different strains. And the next thing that what we are doing is to test them. Of course, we will test them again as antibacterial for antibacterial activities, for their IC50 values. Then we will go for the cytotoxicity analysis, whether they are toxic to the human cells, because salmonella infects the humans, and we will give the drug to the humans. So definitely we will do that. So we have these, because we had last year some funding problems, but now for this year we received the funding and we have purchased these compounds, and we will test it. That is a part of the work. And it's because, as I mentioned, we are facing the salmonella problem again. It's emerging each year. When it comes summer, there are the problems with the water contamination, and the hospital, they are full with the children and the people having the typhoid. So that's one part. The second part, the drug repurposing. Well, that's a very interesting area, especially in the case of corona. We saw that people have tried a lot to find some drugs that are already, for example, remdesivir and the other drugs that you have mentioned. I have gone through that one of your work with the team, and it was very interesting work that have selected some drug as in the case of repurposing for the corona protease enzyme. People are performing two types of studies. One, if you have like a large library of compounds and then go for the screening to get a final set of compounds and then go to the lab and test it. That's one part. The second part, they come in the opposite direction. If there are compounds which are already present, for example, in the market, like the remdesivir and the other compounds against other diseases. So what they do, they select those compounds, make a library, perform the analysis in the lab, and then come and check back the in silico analysis. So it's going both ways, this way and that way, but that totally depends upon the type of study. If you are going for the repurposing, now in the repurposing, of course, we are not going to select thousands and thousands of compounds. We will be selecting like the commercially available, which may be 50 compounds, or which may be 100 compounds, or which may be 200 compounds, and then perform the high throughput screening in the lab, and then go and check as a backup to see if these will be binding with the target or not. Now, the theoretical aspect, it's different from the real aspect. Sometimes the theoretical, it gives you very good results, the free binding free energy, the RMSD value, these things. But when we go to the in vitro studies or to the experimental trials, because we are facing a complete physiological and a different environment where you have the solution, where we have the different solution, the type of cell that you are working inside the cell, the cytoplasm, the metabolites which are present in the cell, so the reactivity, it depends upon that. But if even in my opinion, if it coincides even a little bit, and if it doesn't show a cytotoxicity, and if it gives good pharmacokinetics, the AD-MBT profile, I think that compound it's a good option. In case of your compound, we have to check, for example, whether it's working against the protease of corona. But in the in vitro studies, in the animal trials, for example, infecting an animal model with corona and then giving the drug, how will be the effect of that compound, or doing the in silico work, I guess it will be a bit different, but it is very, very interesting, no doubt, that the drug repurposing is solving many problems in very quick time, instead of taking months and years. You just need a month or two to come up with some good results. Thank you.
Jimena.
Thank you, Hassan, for such an interesting talk. It's not my area of work, but I found it very easy to understand from what you showed of your work. Well, I have two questions actually. First, I found it quite
Tá mudo. Ximena, tá mudo.
It's okay. Está funcionando agora.
better now. Can you hear? Okay. Then I was wondering, how does pharmaco...
Ximena, você tá muda de novo.
Tenta sem o fone.
My connection bunch is not very good. I'll be quick. Can you listen to me now? Can you hear? Okay.
Mas aí para.
Ximena, para, a gente não consegue te ouvir. Melhor você escrever a questão. É melhor escrever a pergunta.
Enquanto ela escreve a pergunta, eu pediria para todo mundo ativar a câmera para a gente tirar uma fotografia de todos os presentes.
Porém, eu, infelizmente, eu como eu CDTS, esqueci a minha câmera em casa.
Bom, quem tem câmera, quem tem. Quem não tem, deixa para lá. Mas eu acho que era bom a gente ter uma fotografia de todo mundo que está assistindo. Isso.
O Marcelo também está assistindo aqui no CDTS e falou que ele está sem câmera, mas ele está conseguindo ouvir.
Que
então,
vergonha.
A gente vai lá para o computador da Priscila. A gente vai lá para o computador da Priscila para poder tirar foto quando for o momento.
Que vergonha, que vergonha.
Bom, enquanto a Ximena...
I think just a complimentary comment. I think even a cell phone can resolve that problem if somebody is missing the camera and the computer.
Yes, it's not easy.
Okay, let me check.
Anyway, but you have a short view from a bird's eye view of our team.
Okay, I got the
and I also see some of your collaborators here. Muhammad Irfan, for instance, hello. And so it's nice to see some of your stuff also here. Ximena, já escreveu?
I got one of her question, and
okay.
she's writing the second question. Okay. So let her write the second question. I'll answer both and try to.
Só ela só quer uma pergunta mesmo.
No, I will have to go on with the first while I write the second.
Okay. So the pharmacokinetic profile, this kind of study, for example, it gives you an idea because each time if you select an inhibitor, a compound against a protein, against an enzyme, against or if it's a bacterial or viral or any, the pharmacokinetics give you the kinetic study of that compound, whether it is if it is absorbed by if it is absorbable, whether it will decompose, whether it will metabolize, whether it is toxic or not. So they have for example, if you go to the Swiss ADME for performing this kind of analysis, or there is another one, ADME T*2 from the Chinese side. These are very good software. So they give you an estimation about your compound, about the molecular weight, the solubility, the lipophilicity, and definitely they have some data analyzed already in the system. So based on the studies they have performed and the information they have present in the system, they give you an idea about your compound. You just select you give the structure of your compound to that program, and it will analyze everything. Or if you don't have the structure, you have some ID or the MOL2 information, because there are different types of the SMILE information. There are different kinds of information for each compound, depending upon the database that you have. You just give them, and it will calculate that study for you. And most of the times, it looks good. Of course, the experimental validation will be required, but this is an initial information. For example, if we are going somewhere, we don't know anything, and we just get a message from someone, it's a good place. So it gives you the preliminary information about these compounds. Okay. It's hard to find studies for drugs in neglected disease. I was happy to see you will work with leishmania. in the future. Parasites are more complex than bacteria or viruses. How different is the approach with the parasite? Well, the approach with the parasites, I'm sure it's because if you go to the parasite studies, you go to the number of chromosomes that are present. If you go see the number of genes and proteins that's present, it's much more. For example, Leishmania, I think it's 30 MB or maybe more or less the size of that parasite. While the bacteria normally it's 2 to 3, 4, or maximum 6, 7 MB. So of course, in that case, what there is a group. There was a group. I'm sure it still will be there in the UFMG, who has performed some functional genomic analysis. She has sequenced a complete genome of the Leishmania, and she combined that with the experimental data. She did, for example, the genome sequencing, all the chromosomes, the genes that were predicted. And then she selected the target using almost the same kind of protocol that I use. And then she selected some inhibitors and went for the in vitro and the lab studies. And it was very, at that time, I guess she published that work in, I guess it was in 10 plus impact factor. I forgot the name of the journal, but that group was working. So it can be applied, but at the moment, most of these softwares and the platform, they are tested on the bacterial and the pathogen and the small genome data set. But if you divide the data set of all these chromosomes and perform for individual chromosome, then hopefully there will be some good results, and definitely there will be software if you are successful in making such a program for the parasite. People will appreciate it because there aren't that much software available for Leishmania or Trypanosoma cruzi or brucei or these kind of infection. So the approach will be a slightly different, but there are studies. I have gone through some of the studies. I'll share sometime with you. It's a group in the UFMG. I forgot the name. I think it's Fernanda, or it was. I forgot. I'm sorry, but there are studies. Oh, it's bad not to hear you, but I believe it was it will be the answer you are waiting.
Any more questions? Sorry, Claudia and Marcos. Well, if there are no more questions, I would ask Felipe. Felipe, você pode parar a gravação agora, tá?
Sim, senhor.
I'm telling Felipe that we should start recording.
No Zoom, mantém no Zoom?
Mantém o Zoom.
Tá.
So that we can hear in the Zoom. Mas não grava o Zoom também não, tá? Deixa só o Zoom aberto.
Sim, senhor.
Okay, I think now, again. Yeah, obrigado. No, I asked to stop the recording because we should feel free to discuss